So like I said, today we will start first with the DNA synthesis. We'll take a look at the various important points that you have to know in DNA synthesis, how the questions can be asked, what are the viva points that you have to know, and what long question or short question can come there. Same will do for RNA, and ultimately same will do for the protein synthesis also. So we'll try to wrap up the thing so that we are able to answer the questions from any of these three which are asked. Very frequently you'll get the question either on the DNA synthesis or you'll get the question on the protein synthesis. So you should be able to write the answer as per a long question. You should be able to draw the diagram, you should be able to write the explanatory notes for each of the steps, you should be able to plan it in phases, and then you should be able to compose your answer. So we'll do the same thing. We'll try to give the structure to our notes so that it is easy for you to reproduce. At the same time, I'll also be talking about the various short notes which are separately asked from this topic. All right, so that is how we are going to go about this. So let us start with the DNA synthesis right away. Let's start with the DNA synthesis. All right, so now we are starting with DNA synthesis. Okay, DNA synthesis. Were there any problems in the test? Were you able to attempt the questions in the test, or were there any doubts or any questions which were not clear in the test? If there are any such questions, you can send them in the message. We'll take them up after the break. Okay. All right. So DNA synthesis. Now, during DNA synthesis, the first biggest problem, which will also be there for transcription, is how to separate the two strands of DNA, because if you do not separate the two strands of DNA, the replication and even transcription cannot occur. In addition to that, there is a problem that you have to separate it from the nucleosomal structure also. We discussed we have to separate it from the nucleosomal structure also. All right. So first thing to note is DNA synthesis occurs in S phase of cell cycle. The first point to note, the DNA synthesis occurs in the S phase of cell cycle, and you should remember the phases. We have mentioned this previously. G0, that is the resting phase, G1 followed by S followed by G2 followed by M. So we are talking about this phase. This is where the synthesis of the DNA occurs, and at the same time you should remember in all these phases we should have the chromatin. So once the synthesis is completed, the DNA will be rearranged as the chromatin. So we are going to synthesize the DNA. This synthesis will occur in the S phase of cell cycle. Now another point which you have to understand is DNA synthesis, which is also known as replication. The term for DNA synthesis replication. So you have to remember that replication is semi-conservative. This is a very important property for the DNA. This is a unique phenomena, that semi-conservative nature of replication. Nowhere in the biological system, when you make a new molecule and you examine the structure, you find half of it is still old. That's what we mean by semi-conservative. I will show you. See, if this is the DNA and you are going to replicate it, then what you will do? First, you will unwind the two strands. So the two strands are now like this, and then you will use each of the strand as a template and synthesize a complementary strand. This is how we are going to do the DNA synthesis. You synthesize the complementary strand. So now we say, okay, this is the new DNA and this is the new DNA. But if you examine the structure carefully, you find half of the DNA is coming from the old DNA. All of it is not brand new. That's why we use this term semi-conservative. Half of the old structure is retained. So replication is semi-conservative. First point to understand is, in the new DNA, one strand is coming from the old DNA, only one strand is being synthesized anew. All right, only one strand is being synthesized anew, and the two strands will act as template for two different DNA. So one DNA will give rise to two DNA particles, two DNA particles will give rise to the four DNA particles. This is because the replication is semi-conservative. The replication is semi-conservative. All right. So the question which lies in front of us when we discuss the replication is from where to start. From where to start? And this question becomes a bigger problem in the eukaryotes, because in prokaryotes it is simple. We don't have the nucleosomal structure, so it is simple in prokaryotes. You just have to separate the two strands of DNA. In eukaryotes it becomes more complicated because we have to resolve the nucleosomal structure also. All right, so let's talk about the prokaryote, then I'll add the points for the eukaryotes. From where to start? This is the primary question. All right, so for this, is there any problem in the relay? Okay, so first primary question which is there in front of us is from where to start. And for this you have to go back to the Chargaff rule that we had discussed. If you recall, we had said that when there is A and T and when there is G and C, how many hydrogen bonds are there between each of them? Between A and T we said there are 2 hydrogen bonds. Between G and C there are 3 hydrogen bonds. So which is easier to break? Obviously it is easier to break the 2 hydrogen bonds. So in the DNA, we look for areas which are rich in AT component. We look for AT rich region. We look for the AT rich region. Why AT rich region? Because it is easier to break the hydrogen bonds between A and T, since they will have only 2 hydrogen bonds, and we say well begun is half done. So if you are able to start the separation easily, you will require less energy, there is more chance of success. All right, so we look for the AT rich region. This is from where the replication is going to start. Why? Because it is easier to separate, and because it is going to be the origin of replication, it is called ORI in short. ORI here stands for, please note, origin of replication. It stands for origin of replication. Now most of your books will not mention this, but please note the number of ORI is not fixed, it is variable, and it depends on the size of the DNA. In prokaryotes the size of DNA is small. The size of DNA is small in the prokaryotes, so a single ORI is enough. But in eukaryotes the size of DNA is very long. For example, if you look at the chromosome number 1, the size runs in 100 million base pairs. It is very very long, and to accommodate such a huge size will need hundreds of ORI. All right, so the number of ORI within a DNA will depend on the size of DNA. Longer is the DNA, more ORI will be required. But at the same time, please note, when I say ORI, it includes both the strands. The 2 strands together is 1 ORI. The 2 strand doesn't mean there are 2 ORI. 2 strands together make up 1 ORI, and the number of ORI will depend on the size of DNA. In prokaryotes typically we have a single ORI. Alright, so we have this AT region we call the origin of replication, and that is ORI, and at ORI certain proteins will come and get attached, typically known as ORE, ORC, or OBP. They are similar names. ORE is the ORI recognition element. ORC stands for the ORI response complex, and OBP stands for the ORI binding protein. For your purpose you can consider them to be similar, the ORE, ORC, OBP. Why? Because they will come and get attached to this ORI, which is the AT rich region. Let's say that here we have the protein complex which has come and got attached. This is the ORE, ORC, or the OBP. Now what this ORI recognition element or the ORI response complex does, it causes the denaturation in this AT rich region. It causes the denaturation or separation of the two strands of DNA in this ORI, that is the AT rich region. Alright, so ORI response element is uh, ORI response complex is formed, and it results in separation of this ORI. So subsequently we have the structure something like this. So in the area of ORI, there is separation of the strands. There is separation of the strands. All right. This is by the action of the ORI response complex. So initial separation of the DNA strands done by the ORI response complex. But remember, the two strands are complementary. They will try to get attached again. They will try to come back and get attached. So once this separation is achieved, we bring in some new molecules. These new molecules are known as SSBP. SSBP stands for the single strand binding protein. It stands for the single strand binding protein. All right. So we have the SSBP, single strand binding protein, which will get attached to the two separated strands, and the function of this SSBP is it will prevent premature re-annealing. See, until unless our process of replication is completed, it will try to prevent the rejoining of this separated ORI strands. So we call it re-annealing. So it will prevent the premature re-annealing. So single strand binding protein will attach to the single strands. Obviously, when the double strand DNA is there, it will not attach, and it will prevent them from rejoining with each other, so that the rest of the function can occur. Now once this separation is stabilized, at this point we have the entry of a very important enzyme which we call as helicase. Now helicase will cause the further unwinding. It will cause the further unwinding of the DNA. The helicase is going to carry out the further unwinding of the DNA. But what you have to understand here, when the helicase comes into play, we will always have a pair of helicases working simultaneously in the opposite directions. A pair of helicase working simultaneously in the opposite direction means at the ORI, once the initial separation occurs, further separation occurs on both the sides simultaneously. All right, I'll try to show it in a picture. So initially there is a small separation like this, right? And after some time, when you try to visualize, what you will find that this separation has extended quite far. Alright, so it looks like an expanding bubble. Alright, a small bubble where you are putting more air, and the bubble is expanding and expanding. So it gives the appearance of a bubble, and because within this bubble the replication is occurring, it is known as the replication bubble. Alright, so further action of helicase and the process of replication gives rise to the replication bubble. So if you examine the replication bubble, let's say this was the midpoint of the ORI, so you will find that whatever is occurring in one half, we'll discuss the details, don't worry, exactly opposite will occur in the other half. Why? Because remember, the two strands of DNA are running anti-parallel. If you go in opposite directions, What was 5 dash to 3 dash one side will become 3 dash to 5 dash in the other side. What was 3 dash to 5 dash on one side will become 5 dash to 3 dash on the other side when you go in the opposite direction. So we get the replication bubble, and if you examine only one half of this replication bubble, it resembles a fork. I'm sure of all of you have seen a fork in the road, or the fork with which you eat. It resembles a fork. So one half of this replication bubble is commonly called the replication fork. I'm telling you this because we are going to focus on a replication fork to understand what is happening in the replication. Alright, when we take the replication bubble, it becomes very complicated. So we'll take one replication fork and we'll try to see what is happening inside this replication fork within the prokaryotes, how the replication is proceeding. Alright. So helicase will cause further unwinding, and after the unwinding, at the same time the replication process will also start, giving rise to what is known as the replication bubble and the replication fork. The replication bubble and the replication fork. Alright, before I go to the replication fork, one very important point. The helicase, although it causes the unwinding of DNA, it also introduces torsional strain, which ultimately results in positive supercoiling. It ultimately results in positive supercoiling. Alright, now this is a very big problem. Positive supercoiling and the torsional strain is very big problem. To understand this torsional strain and positive supercoiling, I'll like you to imagine you using a basic landline phone. I'm sure all of you have seen the basic landline phone sometime in your life. It is not yet so, we are not so beyond the era of landline that you might not have seen it. So if you have ever had the opportunity to use the landline, when you pick up the receiver and put it back on the holder, what you're basically doing is you are introducing a torsional strain, and you will notice that after a few round of uses, the cord which is connecting the receiver with the base, it starts folding upon itself. That is the result of supercoiling. So if you don't relieve the torsional strain, it will ultimately result in supercoiling. Once the supercoiling occurs in the DNA, further unwinding becomes very, very difficult. Alright, because the supercoiling occurs downstream from the unwinding. Alright, so I'll try to show you what is happening. I'm drawing a simplified diagram of the DNA to make it easier to understand. And here what I'll do, I'll make one of the strands as straight. Although remember, both the strands are present as helix in the DNA. I'm drawing one of the strands as a straight strand, and the second strand I'll show as if it is coiled around this strand. Alright, I'll show the second strand as if it is coiled around the first strand. Now here, one thing you have to note, when you consider a certain length of the DNA, the number of times the two strands are wound around each other in this particular length. In this particular length, the number of times the two strands are wound around each other, this is described as what is known as linking number, commonly written as Ln. Ln stands for linking number. So linking number describes the number of times the two strands will coil around each other. So if I consider the total length, linking number in this case will be 3. If I consider a shorter range, let's say I'm considering from here till here, the linking number in this case would only be 2. The number of times the two strands are coiled around each other, that will be the linking number. Alright. So when the helicase acts here, it causes the unwinding in the immediate area, but the loops do not disappear. They are merely pushed downwards. Please understand, when we are separating the upper part, the lower part will become more coiled. That is the supercoiling. The supercoiling will occur. So see this picture when it will become more clear. under the influence of helicase, what is happening? So this is the central DNA strand, and this is the one strand of DNA. So you will notice one coil is totally removed in the upper area, but in the remaining area the number of coils has increased. So the coil which was in this region has disappeared. So this area has uncoiled. This area has uncoiled under the influence of helicase. But when you examine the downstream area, you find that the linking number has increased. Previously, note, in this area the linking number was 2. Now the linking number has become 3. When the linking number increases in a given length, it causes the torsional strain, and when the torsional strain becomes too much, it will cause the supercoiling. Supercoiling is a problem because it will prevent the further unwinding. So we must remove this torsional strain. All right, and to remove this torsional strain, we bring in another protein. This protein which we are bringing in is the topoisomerase. All right, what we're doing here, we are bringing in the topoisomerase. The topoisomerase has two arms. With one of the arms, it will catch hold of one of the DNA strands. It will catch hold of one of the DNA strands. In the second hand, it has a scalpel. So what will do with the second hand, it will introduce a cut in the DNA. It will introduce a cut in the DNA, catch hold of the lower strand. After introducing the cut, it will catch hold of the lower strand and complete a rotation by 360 degree. It will catch hold of the lower strand and complete a rotation by 360 degree. And once it has completed the rotation by 360 degree, the energy which was released during cutting, it is withheld by the topoisomerase, and it uses the same energy to rejoin. So first it will cut, it will rotate by 360 degree and then rejoin the two strands. All right, it will rejoin the two strands. Let's see what happens as a result of this action. What happens as a result of the action of topoisomerase? I'm sorry, this went away from me. Yeah, okay. And let's see what is happening with the topoisomerase action. Okay, we've completed one rotation. All right, so after one rotation, what happens? The unwinding of one coil occurs because that is the free end. From the free end we are doing the rotation, so one coil, one fold, one turn disappears, and after removing that turn we are rejoining the strand. So what you find now, the linking number in this area has been restored to 2. See, previously it was 2, then under the influence of helicase it became 3, and under the influence of topoisomerase it again became 2. So topoisomerase will remove the torsional strain. It removes the torsional strain. So remember, the topoisomerase will remove the torsional strain. All right. I hope this picture that we have drawn is clear and self-explanatory. Basically, what the topoisomerase is doing, it is decreasing the linking number by its action. It is decreasing the linking number by its action. Little bit more about topoisomerase, because the topoisomerase is a commonly asked question which you should be able to answer. What do you mean by topoisomerase? So you can say topoisomerase reverses the torsional strain by decreasing the linking number, and you can draw this diagram that how the helicase is increasing the linking number and how the topoisomerase acts and thereby removes the torsional strain. Okay, so when we come to the topoisomerase, you have to remember two things here. There are several types of topoisomerase, and there are two classes. There are two different things. Class is different and types is different. When we talk about class, we use the Roman numeral 1 and 2. When we talk about types, we use the Arabic numeral 1 2 3 4. All right. So type 1, when I say type 1, it will have the 1 and 2. When I say type 2, it has the type 3 and 4, like this. All right. So I'm talking about the class 1 and class 2. When I say class 1, it decreases linking number by 1, and class 2 decreases linking number by 2, sometimes even more, in one action. Once it will act, then it will decrease the linking number by 1 for the class 1, and it will decrease the linking number by 2 for class 2. Now, for eukaryotes, the rate of replication is comparatively slower, because why? We have to resolve the nucleosomal structure also. Therefore, the rate of replication is slower. But in the prokaryotes, the rate of replication is very fast. All right. So when the faster replication is occurring, faster supercoiling will occur, and to remove that faster supercoiling, we need the topoisomerase 2 in the prokaryotes. It is critical. It is very important for the prokaryotes. It is very important for the prokaryotes. All right. One very important example of the topoisomerase class 2 is the DNA gyrase. DNA gyrase is a very important example. It is the class 2 type of topoisomerase, and it will help in rapid replication. The class 2 topoisomerase may exist in eukaryotes, but it is not critical. Class 1 is enough. I'm not saying the class 2 is not there in the eukaryote, it is there, but it is not important. If you remove it, then also it will not affect the rate of replication, because the rate of replication is slow, which can be handled by the class 1 topoisomerase. But if you block this DNA gyrase in the prokaryotes, then it will become a problem. And we have a whole class of drugs which you'll read next year in your pharmacology session, which inhibits the DNA gyrase, and they are known as the fluoroquinolones. The fluoroquinolones will inhibit the class 2 topoisomerase. Therefore, the side effect profile is minimal. Why? Because they're mainly inhibiting the DNA gyrase 2, which is critical for the prokaryotes, not for the eukaryotes. So these are some extra points you should remember about the topoisomerases. Class 1 and class 2. Class 1 will decrease the linking number by 1. Class 2 decrease the linking number by 2 or even more. Class 2 will support the rapid replication, therefore they are more important for prokaryotes, and we have a group of molecules which can inhibit the DNA gyrase, which is a class 2 topoisomerase, that is the fluoroquinolones. Remember the function of topoisomerase. The topoisomerase remove torsional strain. Till now I've talked about 4 proteins for topoisomerase. Then we talked about helicase, which causes unwinding. We talked about the single strand binding protein, and we talked about the Ori response element. These are the 4 proteins we already talked about. All right. So let's go back to the replication fork. What is happening in the replication fork? Now that you've understood the function of helicase, let's go back to the replication fork. So what I'll do, I'll draw the replication fork, a bigger diagram for replication fork. All right. Please remember, the 2 strands of DNA are always anti-parallel. The second point to remember is the DNA synthesis. DNA synthesis will always occur from 5' to 3' direction. So when the new DNA synthesis occurs, it will occur in 5' to 3' direction. Now, to carry out the DNA synthesis on the second strand, it must occur from 5' to 3'. Alright, and for that you will have to reach 3' end. But once this this is the DNA polymerase. Once this DNA polymerase goes ahead, it will not come back. The complex doesn't return once it moves from one zone of DNA. So the replication of the lagging strand should also be completed at this time only, and therefore it is completed only in parts. It is completed only in parts. You will notice that I am drawing a box at the beginning of each DNA. This box is very important because the DNA synthesis cannot begin from scratch by itself. The DNA polymerase is unable to initiate the de novo DNA synthesis. This doesn't become apparent on the leading strand. I'll show you later. So what we find that on the two opposite strands, the replication occurs differentially. On one of the strands, we find the continuous synthesis. We find the continuous synthesis and we call this strand as the leading strand. The second strand is known as the lagging strand. Why? Because it shows the discontinuous synthesis. The second strand shows the discontinuous synthesis and therefore it is known as the lagging strand. On the lagging strand you can see fragments of nucleic acid. One part is in blue and one part is in red. The red part is made up of DNA. The blue part is actually made up of RNA. These are primers. I'll talk about primers, don't worry. These are primers. Together the two structure is known after the scientist who first described these, and the name is Okazaki fragments. So on the lagging strand we find the Okazaki fragment which is made up of both DNA and RNA. The Okazaki fragment which is made up of both DNA and RNA. So let's talk a little bit about the DNA polymerase and its peculiarity, and then we'll come back to the replication fork. The DNA polymerase has a very important logistical problem. The DNA polymerase, it doesn't matter which DNA polymerase we are talking about, all DNA polymerases will have the same problem. And what is that? They cannot start the replication. I'll try to explain it in a simple manner. Suppose you have two nucleotides and you want to join them. Alright, you want to join them. You tell the DNA polymerase, okay, go and join the two nucleotides. The DNA polymerase will say, no, I cannot do this. Alright, I'll compare it to another scenario. Suppose in your house there are two kids, small kids, very small kids are there, and you're looking for a babysitter. Maybe your elder brother has a twin, he has twin kids and they're very small, maybe 2 years or 3 years old or maybe 4 years old. So you ask a babysitter to take care of two kids, both of them very small. The babysitter will, no, it is not possible. Two small kids, they will keep crying continuously and I'll become mad. I'll not do that. So same thing, DNA polymerase is unable to join the two deoxynucleotides. Why? Because DNA polymerase has limited ability to join, and two free nucleotides will have lot of energy, entropy. Therefore it is difficult to attach. DNA polymerase cannot join two free nucleotides. Now suppose you have one nucleic acid here and another nucleic acid here. Let's say it is the DNA, and you ask the DNA polymerase, okay, go and join these two strands. The DNA polymerase will say, no, I cannot join these two strands. It is comparable to the scenario where in the house you have two elder children, 12 years old, both of them are 12 years old. You tell a babysitter there are two kids in the house, you have to take care of them. He'll say, no, 12-year-old kids, they will tie me in the chair and they will destroy the whole house. You'll come back and you'll kill me. So two elder kids, no babysitter will become ready. So the same problem, the two strands of DNA cannot be joined by the DNA polymerase. What will the DNA polymerase do? Very simple. You must have a pre-existing nucleic acid to which a free nucleotide can be attached. This is what the DNA polymerase can do. You must have a pre-existing nucleic acid in front of which a new nucleotide can be added. This is like you have two kids in the house, one is 10 years old, another one is 3 years old. So babysitter will say, okay, with the help of 10-year-old I can take care of the 3-year-old, both of them will be happy, not a problem. If there are two kids but there is age difference, I can take care of that. Same way, one free nucleotide and one nucleic acid, then the DNA polymerase can act. So you see the problem here. To make this strand at some point in time, you will have to carry out this reaction, but the DNA polymerase cannot do this reaction. How do we carry out this reaction? Because the DNA polymerase cannot do this reaction, we are left with only one recourse, and that is RNA polymerase. But remember, when we say RNA polymerase, this obviously means the product of RNA polymerase will be RNA. RNA polymerase will join the ribonucleotides, it will not join the deoxyribonucleotides, so the RNA polymerase will make the RNA. Alright, so the primer which is synthesized, is RNA in nature. It is synthesized by the RNA polymerase, so the enzyme is called the primase. Primase will synthesize the primer. So you can see the primase is RNA polymerase, and therefore the primer is RNA in nature. DNA polymerase doesn't mind. It says it doesn't matter whether the nucleic acid is RNA or DNA. You have to give me a starting material on which I can add. So using this primer, the DNA polymerase can then add the subsequent deoxynucleotides. Alright, so this primer is what we have seen here. This primer is what we have seen here. The primer is there, and in front of primer, then the polymerase will extend the DNA. This means the primase must travel ahead of the DNA polymerase, because the primer must be synthesized, which the polymerase can extend, and actually it happens in that manner only. The primase is attached to the helicase. So here we have two proteins, the helicase to which the primase is attached. Means they are traveling together. I will not say attached. The helicase and primase are traveling together. Helicase and primase are traveling together ahead of the polymerase. They are traveling together ahead of the DNA polymerase. They are traveling together ahead of the DNA polymerase. So you can ask the question, sir, what about the primer here? Is there no primer in front of the leading strand? We'll come to that, but first let me show you how these segments are resolved, because once the replication is completed in the DNA, we don't find the RNA. It is a complete continuous DNA. There is no talk of leading and lagging strand in the final output. Alright, I had drawn this final output here. Here we have not talked about any leading or lagging strand. Both strands are complete DNA. Here also both strands are complete DNA. So how this segment where we have the RNA is resolved, let me first discuss that, and then I'll come back. What happens to the primer on the leading strand? We'll come back to that in a minute. Okay, remember the first DNA polymerase which is acting in the prokaryotes is the DNA polymerase 3. Immediately after the helicase and primase, it is the DNA polymerase 3 which is the main replicating DNA polymerase is the polymerase 3. Now here, to resolve this RNA fragment, we have the entry of polymerase 1. Polymerase 1 will come here and it will resolve the primer. I'll show you how the primer is resolved. So let's say this is the strand with unfavorable orientation, that is 3' to 5', and it will have the lagging the Okazaki fragments. So I'll draw the RNA with blue and the DNA with red. Okay, So this is the primer, and this is the DNA fragment. Okay, another primer, another primer, and another DNA fragment. So basically what we have in front of us are two Okazaki fragments. We have two Okazaki fragments. I'll color them differently so it is easier to appreciate. The Okazaki fragment 1, and this will be Okazaki fragment 2. Alright, obviously the colors will not stay when we are discussing. So okay, so I'll color them at the bottom. This is one segment and this is the second segment, overlapping segments. Okay, so I told you here we have the entry of the DNA polymerase 1. Now polymerases have the dual action, they have the proofreading function also. Proofreading function is also there. Typically the proofreading function is in 3' to 5' direction. I'll write it here. The proofreading function normally is in the 3' to 5' direction, but for polymerase 1 we have an additional 5' to 3' proofreading function. We have an additional 5' to 3' proofreading function. Proofreading function means if there is an incorrectly placed nucleotide, it can be removed. Alright, proofreading means you are checking whether the replication is occurring correctly or not, and if it is not occurring correctly, then you can remove that particular nucleotide. So when the polymerase 1 comes in and it examines this particular segment, alright, so we are trying to make the DNA. So here the deoxynucleotide should be there. When it examines it says, alright, these are not deoxynucleotides, they are ribonucleotides. So as per the proofreading function, these nucleotides are incorrectly placed. So what it will do? It will remove the nucleotide. Let's say it first removes the first nucleotide at the 5' end. Now remember, I have told you repeatedly, this polymerase will bring in the deoxynucleotide, but a new nucleotide can only be added at the 3' end, not at the 5' end. Recall when we were doing the nucleotide structure, there I had described when the two nucleotides join together, the new nucleotide always comes at the 3' end. There is no provision for joining the nucleotide at the 5' end. So the deoxynucleotide has come, but it cannot join here. Here it cannot join because this is the 5' end. It has to join here only, that is at the 3' end. So this deoxynucleotide is attached to the Okazaki fragment number 2, not to 1. Similarly, the next ribonucleotide is added, removed, replaced by the deoxyribonucleotide and joined to the next Okazaki fragment. The ribonucleotide is removed, replaced by the deoxynucleotide. The last ribonucleotide is removed, last replacement by deoxyribonucleotide. This is what the DNA polymerase 1 will do. It will replace the primer. So what you can see here, polymerase 1 has replaced the primer. Now we are left with two DNA segments. Remember, we had said the DNA polymerase will not join two segments. The DNA polymerase will not join the two segments. To join these two segments, you will have to bring in yet another protein. To join these two segments, we have to bring in yet another protein, and that protein is DNA ligase. DNA ligase joins the Okazaki fragments. It joins the Okazaki fragments. Alright. So the primer is replaced by the polymerase 1, but the joining of the two Okazaki fragments is done by the DNA ligase. So replication by DNA polymerase 3, removal of primer by the DNA polymerase 1, and joining of the Okazaki fragments by the DNA ligase. Once you have understood this, let's go back to the replication fork where the picture will become clear. Remember, I told you we have what is called replication bubble. We have taken only one half of it to understand, that is the replication fork. So whatever you are seeing here has a copy somewhere else, not far, here only. Right? So what was leading strand on one side becomes the lagging strand on the other side. What was lagging strand on one side becomes the leading strand on other side. Now imagine there is an Okazaki fragment here. The RNA is there. As soon as the first Okazaki fragment is formed on the lagging strand on the opposite side, with the help of the Pol 1, with the help of the Pol 1, it will replace and fill in the gap along with the DNA ligase. The same thing will happen here also. So whenever you look at one replication fork on the leading strand, we don't find or we don't describe the primer, because by the time we have reached a little bit ahead, that primer has already been resolved with the help of the polymerase 1 and the DNA ligase. So on the leading strand we don't describe the primer, but on the lagging strand we have to describe the primers and the DNA which form the Okazaki fragment. Alright, so for leading strand we have the continuous synthesis. For the lagging strand, we have discontinuous synthesis where we find the Okazaki fragments, Now, in addition to this resolution of the Okazaki fragment, there is a problem when you reach the end of the DNA. When you reach the end of the DNA, there is a problem. Let's say this is the end of the DNA, and this is the last Okazaki fragment. Sorry, draw it a little bit higher. Okay, so this is the last Okazaki fragment, right? All right, so what will happen? This Okazaki fragment will extend itself and replace and finally join with the next Okazaki fragment. But what will happen to the primer part of the last Okazaki fragment? Remember, it is RNA, so it has a short half-life. You have a very limited amount of time to replace this primer part. How are you going to replace this primer part? How is the primer of the last Okazaki fragment replaced? Please quickly answer. How is the primer of last Okazaki fragment replaced? I'll wait for a few seconds so that you get the question and then you can reply it. How is the primer of last Okazaki fragment replaced? I'll write it here. Primer of last Okazaki fragment How is it replaced? This is a very conceptual question. Let me see if anyone can answer it. Primer of last Okazaki fragment, how is it replaced? It has to be replaced because it is RNA in nature and RNA is not a part of the final DNA structure. Anyone who can tell me the answer? How is the primer of last Okazaki fragment replaced? I'm waiting for answers on the Telegram group. How is the primer of last Okazaki fragment replaced? Please note, you are late. The RNA is gone. You didn't give the answer in time. The RNA is gone. RNA is a very short half-life. You did not come up with a technique to replace this primer and now it is gone. And this is what happens. The primer of last Okazaki fragment can't be replaced because we don't have a Okazaki fragment behind this. If we don't have Okazaki fragment, which DNA are you going to extend? There is nothing behind it to extend. What are you going to extend? Where are you going to replace the RNA by? Because the polymerase, DNA polymerase can only extend a pre-existing nucleic acid. There is nothing behind this Okazaki fragment. What are you going to extend? Nothing. And please remember, it cannot be replaced. This is the reason why, if you recall, I had told you in the, what happened? At the end of DNA, one strand is always shorter than the second strand. This is because the primer of the last Okazaki fragment cannot be replaced. You recall we had said, I told you when we do the replication, then it will become clear why the one strand is shorter than the second strand. The reason is the last primer cannot be replaced. It results in a difference in length of the two strands. It results in a difference in length of the two strands. Basically, what is happening? What is happening here? A small amount of telomere is lost. A small amount of telomere is lost. Don't worry, I did not expect you to be able to answer this question because in most of your UG books this part is not very clearly mentioned. So please understand, a small amount of telomere is lost every time the replication occurs at the 3' end. Every time some amount of telomere is lost. As a result, after a series of replication, all telomere is lost. And remember, what is the function of telomere? It stabilizes the end of DNA. It prevents from apoptosis. So once all the telomere is lost, the apoptosis will occur. Alright, this means a cell can divide only a certain number of times. After that, if it divides further, then the cell will die. This is particularly true for somatic cells. Somatic cells divide only a limited number of times. Somatic cells divide only a limited number of times, and this limit is known as Hayflick's limit. A somatic cell can divide only a limited number of times. This is known as the Hayflick's limit. Alright, somatic cells will divide only a limited number of times, which is described as Hayflick's limit. Hayflick's limit will vary from species to species. It is a property of a species. For humans, the Hayflick's limit is approximately 70 divisions. For humans, the Hayflick limit is approximately 70 divisions. After 70 divisions, the cell will die. Now, this can't be true. Strictly speaking, it can't be true for all the cells. Why it can't be true for all the cells? If it were true for all the cells, then the number of students sitting in the class looking like me will be large. Looking like me matlab there is nothing on the head, there is no hair here, because the hair continuously keeps growing means the cells are stacking one after the other, the cells are stacking one after the other, and this continues throughout the life. If you look at the size of one cell hair cell has a size of 20 microns. All right, 20 microns means for 1 centimeter we need 500 cells. For 1 centimeter we need 500 cells. 1 millimeter will require 50 and 1 centimeter will require 500 cells. And in one month typically we get 3 to 5 centimeter, sometimes even 10 centimeter of hair growth. 3 to 4 inches the hair will grow. Maybe let's only 5 centimeter. Even if it is 3 centimeter, it will mean 1500 cells stacked one after the other. And here we are saying how many? 70 divisions. So we can see the Hayflick's limit is not true for all the cells. It is true for the somatic cells. Somatic cells have a limited number of divisions, but it is not true for all other cells, meaning there is a mechanism to bypass this Hayflick's limit. There is a mechanism to bypass this Hayflick's limit. How do you bypass the Hayflick's limit? The answer is very simple, very elegant, and all of you know this. We can bypass the Hayflick's limit by an enzyme complex known as telomerase. Telomerase will regenerate. It regenerates the telomere. So whatever shortening occurs, that is recovered. In addition, extra telomere is synthesized by the complex telomerase. Now telomerase is a very big complex. It contains large number of RNA and the proteins, but out of that whole complex there are two which you must know. The telomerase two important components are what is known as TERC. TERC stands for telomerase RNA copy. What it basically is, RNA which is complementary to telomere. Remember, the telomere has a very specific sequence. We had said it has a self-complementary repeating sequence. So we are talking about an RNA which is complementary to telomere sequence. RNA which is complementary to telomere sequence. And the second component which you should know is the TERT. TERT is telomerase reverse transcriptase. Telomerase reverse transcriptase. What does the telomerase reverse transcriptase do? Creates DNA from RNA template. It creates DNA from RNA template. So these are the two primary molecules which are required to regenerate the telomere. The two important molecules which are required to regenerate the telomere. Try to understand very, very simple process which is occurring. This is the new DNA. The lower one in black is the old DNA. The lower one in black is the old DNA. So once the replication complex has completed its work, the telomerase will come in. The telomerase will come in. the RNA copy will align itself with the overhanging edge. It will align itself with the overhanging edge, and you will notice it is longer than the overhanging edge. Alright, so this is, remember, RNA. Using the enzyme reverse transcriptase, what we do, we extend the parent strand. Remember, this was the 3' end. This was 3' end. So we can add the nucleotides at the 3' end. Once the extension is complete as per the RNA template, the complex will go away. Alright, and it will rejoin some distance ahead. Again, the RNA will align with this overhanging edge. The reverse transcriptase will come in and add more nucleotides. This process is repeated 4-5 times. This process is repeated 4-5 times. Alright, this process is repeated 4-5 times, and you can see that the parent strand has been extended significantly. Now, please keep it in your mind that the difference in length is still there. But what you have to note, previously our synthesis of the new strand was finished here, and the original DNA was getting over here. But now even the shortened strand is bigger, or you can say longer, than the older strand. In this way, the telomere has been regenerated and extended. By the action of the telomerase complex. Here you should know tissues where telomerase is physiologically active. The stem cells can be hematopoietic stem cells. They can be hepatic stem cells. They can be splenic stem cells. Now we also have the concept of neural stem cells. We have the concept of respiratory stem cells. So stem cells wherever they are present. Similarly, the stem cells in the hair follicle. The male germ cells. Please note, not the female germ cells, the male germ cells. Why? The female are born with a full complement of oocyte. In the lifetime, the meiosis does not occur, so they don't require the telomerase. And the epithelium of skin and GIT, they are also continuously growing. And one very important additional name which you should not forget is the cancer cells. The cancer cells have activated telomerase, which helps them to bypass the process so that they can keep dividing unlimited number of times. So cancer cells also have activated telomerase, irrespective of their origin. They have activated telomerase, which helps them to bypass the apoptosis. Alright, so this is about the telomerase, how it helps to counter the ill effects of the loss of telomere, and how the stem cells can keep dividing unlimited number of times. Let's go back to our replication. So here what is happening, the replication has been completed. All the Okazaki fragments have been replaced. The last primer of the last Okazaki fragment cannot be replaced, results in shortening of the DNA on the lagging strand. The 3' end is shortening of the DNA. Once the replication is completed, don't forget, the DNA reorganizes as chromatin. Remember, this replication is occurring in the S phase. In the right in the beginning, I told you in the G0, G1, S and G2 phase, the DNA must exist as chromatin. So once the replication is completed, the DNA will reorganize as chromatin. This is a very important point to keep in mind. What happens to the DNA once the replication is completed? It reorganizes as chromatin, not as chromosome. It reorganizes chromatin, not as chromosome. The chromosome will come only in the M phase. Take a look at the two important enzymes in the telomerase, two important molecules in the telomerase complex. The two important molecules in the telomerase complex, the telomerase RNA copy and the telomerase reverse transcriptase, they will be responsible for carrying out the main process. The RNA copy has a very peculiar feature. It is complementary to the telomere sequence. Because it is complementary to the telomere sequence, it can get attached to the telomere. It will get attached to the telomere. Let's see. I'll discuss the telomerase complex again. Although this is a bonus topic, it is less likely to be asked in your exam. How the telomerase acts is less likely to be asked in your exam. Okay. This is the end of DNA after the replication has been completed. You can see one strand is shorter than the second strand, and this is where the telomerase will come in. The telomerase will come in, and we said we have the RNA copy which is complementary to the telomere. RNA copy which is complementary to the telomere. Because it is complementary to the telomere, with the help of Chargaff base pairing, it can attach itself. It can attach itself to the overhanging edge. This is the parent DNA strand, and we are talking about the 3' end. All right, and then the reverse transcriptase comes. What is the function of reverse transcriptase? Using the RNA as template, it can synthesize the new DNA. Using RNA as template, it can synthesize the new DNA. This is how the parent strand is extended. Once the parent strand is extended, the replication machinery can further synthesize the complementary sequence on the parent strand. So we have an extension of the telomere even beyond the size of the parent DNA strand. Extension of telomere even beyond the size of the parent DNA strand. I hope this makes it more clear. In a more simplified picture, I have tried to show you how the telomere is extended beyond the size of the parent strand. The two main molecules which are playing the role are the telomerase RNA copy and the telomerase reverse transcriptase. Obviously, there are a very large number of proteins which are also coming in, but you are not concerned about their function. They have the helping role. They are supporting actors. The main role is acted upon by the RNA copy and the reverse transcriptase. Syllabus will be what we cover today and what we covered on last Saturday. All right, or last Friday. Whatever we covered on last Friday and what we are covering today, all of that will come in your grand test. I hope the process of replication is clear to everyone. The process of replication is clear to everyone. Please understand that what we have discussed as replication is primarily what occurs in the prokaryotes. In eukaryotes, the whole picture is not yet clear, but it is quite similar to what happens in the prokaryotes. Here, I would also like to compare the DNA polymerases in the prokaryotes and eukaryotes. But before that, let's summarize the function of the different proteins. I'll just write the names because the functions we have already written. What are the various proteins that you should know about? We started with the ORI recognition element or the ORI binding protein. Alright, we already said it causes separation of the origin. Then we brought in the single strand binding protein. Remember, we had written it prevents premature reannealing. We have written that previously. It prevents the premature reannealing. Thank you. Then we have the helicase. It causes the unwinding of DNA. And then we have the topoisomerase. Topoisomerase removes the torsional strain. After that, we had the enzyme primase, which synthesizes the primer. The DNA polymerase 3, which is responsible for the primary replication, followed by DNA polymerase 1, which replaces the primer. DNA polymerase 1 replaces the primer, and the DNA ligase, which joins the Okazaki fragment. These are critical for replication. Telomerase is not critical for replication. I have not written the name telomerase here. Remember, telomerase is not critical for replication. It is required only in non-somatic cells. In somatic cells, there is no role of telomerase, meaning it is not critical for replication. So these are the proteins, and you should be able to write the function of these proteins, and you'll get very frequently the short note on topoisomerase. You'll get the short note on the DNA polymerase. All right, so we'll talk about it. Sometimes you can get the question on the DNA ligase also, but primarily you get the question on the topoisomerase and the DNA polymerase. So let me quickly talk about the DNA polymerase, and then we'll compare the prokaryotic and the eukaryotic polymerases. Yes, I told you the telomerase is responsible for the immortality of the cancer cells. It helps them to bypass the process by keeping the telomere intact. That's why telomerase is a primary target of research to counter the immortality of the cancer cells. Till now, we have not yet achieved success or a breakthrough in that research, but it is a topic of hot research, how to target the telomerase enzyme in the cancer cells and at the same time not hit the normal cells where the telomerase is active. We are expecting that in the near future we get a breakthrough in this, which will help us in very specific targeting of the cancer cells. Okay, quickly I'll talk about the polymerase. Part of it I've already told you. When we talk about the DNA polymerase, here you have to keep in mind three things. First is the property of chain elongation. First is the property of chain elongation. Second is the property of processivity. These two are similar, so I've written them together. Chain elongation is basically the rate at which the nucleotides are added, and therefore it is written as nucleotides added per second. It is written as nucleotide added per second, whereas the processivity is the total work done, and therefore it is simply written as the total number of nucleotide added during one attachment. What you have to understand here, the DNA is very long. We have two strands of DNA where the replication is going on. The DNA is very long, so the polymerase attaches here. And it starts carrying out the replication, but because the DNA is very long, the same polymerase is not going to replicate the whole of DNA. After some time it will get off, it will dissociate. All right, so the processivity is the total number of nucleotides which have been added during one attachment. This is the total number of nucleotides which have been added, whereas the number of nucleotides added per unit time, that is the chain elongation. Please note both the activity must occur in 5 dash to 3 dash direction. We already said the nucleotide is always added at the 3 dash end, so the chain elongation always occurs in the 5 dash to 3 dash direction. So we have the chain elongation and processivity, both are oriented in 5 dash to 3 dash direction. One is the rate and the second is the work done. The third point is the proofreading activity. This I've already mentioned. The proofreading activity checking for attachment of correct nucleotide. Checking for attachment of correct nucleotide. Now please note, we said that the direction of synthesis is 5 dash to 3 dash. So imagine I've added a nucleotide here. Now I want to check. I have moved forward, but I want to check whether the addition is correct or not. So I have to rotate. Now my direction has become 3 dash to 5 dash. So remember, normally the proofreading activity is 3 dash to 5 dash. The proofreading activity is normally 3 dash to 5 dash. In all the polymerases, we have the proofreading activity in 3 dash to 5 dash direction. In polymerase 1, there is an additional 5 dash to 3 dash proofreading action. This is an extra. In addition to the 3 dash to 5 dash proofreading activity, we have an extra 5 dash to 3 dash proofreading activity, particularly in the polymerase 1. It will also be there in the counterpart in the eukaryotes. So these are the three properties of the polymerase. So when you are getting the question on DNA polymerase, you will quickly write these three properties and then you will draw the table comparing the prokaryotic and the eukaryotic polymerases. I'll quickly draw that table comparing the prokaryotic and eukaryotic polymerases. So we have this very small table, prokaryotes, the eukaryotes, and in middle I'll write the function. In prokaryotes we have the polymerase 1, DNA polymerase 2, DNA polymerase 3, and a protein called DNA G. Polymerase 1, we have already said, has two important functions. Number one, it replaces the primer. In addition to that, it has the repair function. Polymerase 2 primarily has the repair function, whereas polymerase 3 is the processive enzyme. Processive enzyme means the enzyme which can carry out the polymerization at the fastest rate. It is the enzyme which can carry out the polymerization at the fastest rate, and the DNA G is the primase, an independent protein in the prokaryotes. The primase is an independent protein in the prokaryotes. Whereas in the eukaryotes, the replacement of primer is done by the DNA polymerase beta. The processive enzymes are the delta and epsilon. The processive enzymes are delta and epsilon. The primase is a subunit of, means it is not an independent protein, it is a subunit of polymerase alpha. So what you will note, I have mentioned alpha, beta, delta, epsilon, but I have not mentioned gamma. I have not mentioned gamma because gamma is not present in the prokaryotes. It is the mitochondrial DNA polymerase. In eukaryotes, the DNA polymerase for mitochondria is also coming from the nuclear DNA. The mitochondrial DNA polymerase, that is the polymerase gamma, is coming from the eukaryotic genome. One very important peculiarity for the mitochondrial DNA polymerase you have to remember is it has poor proof reading activity. If you recall, we had said the rate of mutation in the mitochondrial DNA is 5 to 10 times higher than the nuclear DNA. Reason being, the polymerase which is acting here has a poor proof reading activity. It has a poor proof reading activity. Alright. So when you get the question on mitochondria, first we'll quickly describe, sorry, on the DNA polymerase, first we'll quickly describe that DNA polymerase is primarily responsible for creating the new DNA, that is polymerization. The main properties of the DNA polymerase are the chain elongation, the processivity, and the proofreading activity, and then you'll describe what we have written. Then ultimately you can compare the polymerases in the prokaryote and in the eukaryote, what are their specific functions. About repair, all the polymerases in the eukaryotes are generally involved in repair. It is not assigned to a specific polymerase. All the polymerases in one way or another are involved in the repair mechanism with the help of the big complexes. So we don't specifically mention a polymerase for the repair. So this is about the replication process. One small thing which is left here is the DNA damage and the DNA repair mechanism. So we'll take a short break here. After the break, we'll quickly talk about the DNA damage and DNA repair mechanism, which is another very important question, very frequently asked in your exam. In the clinical aspect, either the name of the disorder is given or the question clearly says describe the DNA damage or describe the DNA repair mechanism. So we'll take a look at the DNA damage and the DNA repair mechanism after this short break. Alright, so let's take a short break and come back in 15 minutes, have a cup of tea, coffee, refresh yourself, and then we can go on with the DNA damage and the DNA repair mechanism. Sure. Okay. In the meantime, if you have any questions, please put them in the group, and after the break I'll take up. If you have any doubts, any question, any part you want me to repeat again which you've discussed till now, then I'll take up all those.